about synchrotron radiation-based analysis at nanomaterial and bio interface. My talk includes 6 parts. In the past 20 years, thousands of nanomaterial have been used in industrial field, consumer goods, biomedical fields, due to the because nanomaterial have excellent physical and chemical properties. The nanomaterial have small size, high surface activity, tunable surface and shape, and very good physical properties. These properties may endow their very excellent performance in the application. From another aspect, the properties such as high surface activity may trigger some safety and hazard concerns. So, my interest is that how is to answer the fundamental question: how nanomaterial inter with environment and our biology system, how we capture and detect their interaction with our biology system. Because the biology system and environment is very complex. So, for example, if we want to use the nanomaterial as a powerful nanomedicine, we still have a long way to go. Because one reason is that our biology system is very complex. Another is that we still lack proper analytical method to study how nanomaterial interact with our biology system. From this roadmap for therapy nanomedicine, we still need 30 years to go to obtain an excellent nanomedicines. We can see that we need to know many information about how nanomaterial interact with our tissues, such as with tumor, liver, spleen, kidney, blood, and other system. Next, we need to know how nanomaterial interact with small animal and a bigger animal, a large animal. Then we need, based on this information, and modulate the properties of nanomedicine to have a good performance, not only good performance, and also have a good safeties. So that costs a long time. During the study, we should take this take some powerful analytical method to study how they interact at different levels and know the important knowledges at the different levels. Then we based on this knowledge to design the nanomedicine, have a good performance and good safety. So that's the reason why we need no time to design and develop develop good nanomedicines. As we know, the biology system is very complex. For the whole body of human being, when nanomaterial is exposed to human being, they will be absorbed and distributed in different tissues and organs. And then some tissue organ like liver and spleen, material may stay there for a long time, or part of them may be metabolized and secreted by the urine and feces. A small part may remain in the tissues for a long time. So during the interaction, there are some complex reaction happened. Due to the one reason is the complex of system, biology system. They include many kind of molecules, cells, and tissues, and the concentration of proteins and molecules are heterogeneous with different concentration and different distributions. Moreover, the interaction is a dynamic process. The one nanomaterial interact with cells and with the proteins. So all the things are so complex that make the study for nano whole body study very complex. Even for the simple cells, the nano and cell interaction is also complex. For example, when nanomaterial interact with cells, first they absorb the proteins in the environment, and they form some nano protein coronas on the surface. The complex may be recognized by cell receptors. Then they translocated to the endosome and nanosome. In the organelles, they will be some of nanomaterial remain in the organelles. Some of them may escape from the endosome or nanosome, may enter the cytoplasma, or may target some organelles like a nucleus and a mitochondria. The rest of them may be removed by cells. So from the whole picture of the interaction, the nanomaterial change the localization and the physical and chemical conditions all the time. So if we want to know what happened to the cells during the interaction, we need to confirm what happened to nanomaterials. This is maybe the reason caused some biology response or some biology effect of some effect. So to know the question, we needed to answer several questions. First, how nanomaterial interact with the environment. One is the protein, another is the lipid. Yeah, protein is outside of the cells. Inside the cells also have include some proteins. So nanoprotein is the basic interaction during the nano-bio interface. Another thing is the nanolipids. When nanoparticle is going inside cells, the cell membrane is the first barrier. The second barrier is their organelle membrane. So nanolipid interaction is also important interface. The third one is the chemical reactions at the surface of nanomaterials. Because although we know the endosome and nanosome is acidic environment, and there are other redox enzymes inside the cells. So this suggests that nanomaterial may involved in some chemical reactions. So so we need to know what happened at the chemical reaction for nanomaterials inside the cells. From the whole picture, we needed to answer several questions at the different levels. One is protein, lipid, another is inside the cells. Because the complexity of the interaction, we need different ways to capture the interactions. One way is that we need to know that when that particle go to the specific microenvironment, what kind of interaction at the interface of nanomaterial, because nanomaterial surface play major roles when they cause the toxicities and induce some effect. The surface is very important. So we need to know what happened to the surface of nanomaterials. Second is when nanomaterial go inside the cells or in the body, we not want to know their distribution. So imagine it is important to know the distribution of nanomaterial. Third one is nanomaterial is not inert. Sometimes they are chemical reactive. So we have to know what kind of chemical reactions to the nanomaterials and what the result of the reactions and the outcome of the biology outcome of the reactions. That means that we need to capture the chemical forms and changes of nanomaterial inside the biology system. To answer these fundamental questions, we need to use some powerful techniques. As we know, X-ray based techniques is excellent tools to study the elements. They can identify elements in complicated system they can also detect the electron densities to distinguish the different molecular and different metals. So that suggests that they can distinguish the chemical forms and the chemical valence of elements. So the one that X-ray have excellent ability to penetrate the samples. They show high energies. They can penetrate samples with much higher depths. But for visible net and electrons, the ability is weak. They hardly need to penetrate cells. So that means that X-ray can be used to do 3D imaging for single cells. So X-ray is powerful to get not only to obtain imaging information, we can also to do structure and a structure for nanomaterials and molecules. So this is how to get a high quality X-ray. Synchrotron radiation is a facility that can provide a very high brilliant and high quality X-ray. That energy X-ray have energy resolved, energy tunable, and monochromatic. So this is a very high quality X-ray can be combined with some methods such as spectroscopy and scattering and images. So with this method and high quality X-ray, we can do many studies for structure and distribution of elements and the dynamic of molecules, also for nanomaterial and nanomolecular interactions. They can also to imagine the elements of nanomaterials inside the cells and inside the single cells and the tissues. So we make a summary about these techniques for nano and bio interface. Here we want to highlight the nano-bio interface. We highlight the interaction and interface at the 3 levels. One is nano and microenvironment interface, such as nanomaterial with interaction with protein and lipid. That is the very directly interface. Second is the chemical reactions of nanomaterials inside the cells or inside the tissues. So the second interface is what happened to the nanomaterial, to the surface of nanomaterials when the chemical reaction happens. So we capture these chemical reaction at the single cell level and at the animal levels. To know the processes, we developed several kind of methodologies. One, for example, for liquid interface analysis, we can use the synchrotron radiation CD and FTIR to detect protein structures when before and after the interaction with protein. Second, we can use the X-ray absorption spectroscopy to detect the binding site of nanomaterial with some proteins. When the protein form the hard corona on the surface. Third one, we can use the X-ray reflectivity to detect how nanomaterial interact with the lipid to detect the lipid structures after the absorption. So the fourth one, we can use the X-ray absorption structure to detect the chemical interaction for nanomaterial in some chemical environment. For example, netsome or ensome environment. What kind of degradation and the chemical states happened to the nanomaterials. So this is the first liquid interface analysis. Another is for imaging techniques. imaging technique can detect the in situ chemical reactions of nanomaterials. For example, we can use the X-ray fluorescence. This method can use the 2D scanning for element imaging. They can achieve a high resolution and have a high sensitivity to detect the elements inside the cells or tissues. For another scanning transition X-ray microscopy that also can do 2D or 3D element images, and they can have a high resolution and high sensitivity. They can also detect the distribution of different elements with different chemical forms. For example, for iron valence 2 or valence 3, using these techniques, we can see the different distribution of the 2 valence of irons inside our cells. So this is powerful for detect the chemical forms distribution. The third one is the transmission X-ray microscopy. This technique is very powerful to get a 3D imaging for elements, because this technique uses X-ray to transmit the samples. Then the sample rotates for 180 degrees. Then we can reconstruct the figures to get a 3D imaging for the samples. We are using the method to detect the distribution of nanomaterial inside a single cells, a single bacteria, a single hazard particles. So basically, these imaging techniques and liquid interface analysis, we study different nano-bio interface structure and informations. And this is our high resolution X-ray imaging techniques, X-ray, XRF, TXM, and STICSOM and CDI. This method have some advantage, then have more advantages than the fluorescence imaging and the non-labeling imaging. For example, they are sensitive for elements. Elements-specific imaging can be obtained by all of them, but they no need to label the particles, and also they are high sensitive to reach this high femtogram per micrometer square. So they can also to study the in situ chemical reactions for the cells. But for the fluorescence imaging, they also have some advantage for living cell or living sample imaging, but the limited is that the low resolution and will be need some labels. They cannot to detect the chemical reaction of nanomaterial inside cells. For other non-labeled imaging, for example, TM and other techniques, they needed to they had a need to penetrate the samples and cannot get the in situ information. So with this advantage, we will introduce detailed examples for how the techniques are used in nano-bio interface studies. The first example is that about nanoprotein absorption and interaction. As we know, when before net particle interact with cells, they were first interact with the protein around the cells. Some protein have a firm absorption on surface. Some have a loose absorbed on surface. From the absorbed protein called the hard corona. Although we know the hard corona play a very important role to prevent the toxic surface of nanomaterials, but the structure is not clear. So when if we can reveal the binding structure for nano and protein interaction, we could better understand how nanomaterial, how protein corona cause some biology effect and trigger some effect like inflammation, like signal pathway activations. So to answer the question, we develop a new method. We based on X-ray near edge spectroscopy. The techniques can be combined with molecular dynamic simulation. This method can be detected the chemical information of the elements. They can detect the chemical speciation, chemical coordination of the elements. They can also serve as the element fingerprint. So based on this advantage, we can use this method to detect different chemical forms of sulfur inside the albumin. The albumin have different kind of before and after interaction. The albumin, the sulfur chemical form changes. For example, at the beginning, they have a disulfide of sulfur. Then after their interaction, the disulfide will break and into the metal. If the intervals the metal element nanoparticle, they will form the metal sulfur bond, and different kind of sulfur have different spectral fingerprint. So based on this fingerprint, we can calculate what the ratio before and after of the protein interact with the gold particle. They can be used to quantify the sulfur chemical forms. Based on the quantification information of that, we can detect how many disulfide break. Then we can use the MD simulation. The simulation can detect the actual location, location of the protein interact with the nanoparticle. Based on these two ways, we can detect how they interact at the surface of the gold. So that is a very good way to quantify protein and nano interact at the interface. Except that that we can also study other nanomaterial, such as cell nanoparticle. Cell nanoparticle can also with some proteins. The MT is a protein that can interact with the metal ions, being quench the ions via their cysteine amine acids. So use this similar method, we use the SRCD, and thus we can detect how the protein change the structures and show the binding site of the proteins. Moreover, we can also use the SRCD to detect the protein signal structures. The signal structure is SRCD is more powerful than traditional CD because the CD can get a much lower wavelength than the traditional CD. Traditional CD can only from the 190, and this SRCD can reach the 160 nanometer. So between 160 and 190, there are many detailed signal structures. So based on this detailed structure, we can calculate and fit the structure between after and before and after interact with proteins. They can get a more detailed structure information. Moreover, for SRFTIR, we can also detect the interaction with the proteins. So based on this SRCD, we study this 2-dimension the norm disulfide net sheet interact with four kind of proteins. We can observe that the protein changing the second structure after the absorbed on the surface. Based on the structure, we can conclude how nanomaterial trigger the inflammations. So the structure is very important to show the mechanism. Next, we use the liquid surface scattering to study how nanomaterial interact with the lipids. This X-ray reflectivity and G-source are two kind of powerful scattering method to study what happened to nanomaterials and lipids on the surface of liquid. These two techniques can be used to provide high sensitive and high resolution information than the conventional method. For example, if we study nanomaterial, the interaction between nanomaterial and lipid, we use the electron microscopy and net microscopy and AFM. But this method, most of the time they need dry the environment. Also, the imaging need some dyes to sustain the samples. They are not the real information. So X-ray based on liquid techniques can provide the in situ liquid environment structures. So the structure include the thickness of the lipid and the directions of absorbed nanoparticle and the amount of the nanoparticle on the surface of lipid. Based on this information, we can study how nanomaterial structure or surface affect the lipid structures. For example, we studied the gold lead rod. This gold lead rod have different coatings. One is the surfactant coated C-type gold lead rod. They have a bilayer structure on the surface of gold, but the bilayer structure is not stable. Another modification is the polyelectrolytes. This modification is based on C-type and another negative surface. So by bilayer assembling, this polyelectrolyte maybe go to the surface of the net rod. So to compare these two kind of coating and the effect on the cells, we found that the surfactant gold lead rod may cause the cell death and damage the cell membrane, but the polyelectrolytes gold lead rod, they are safe to cells and didn't cause cell death. Though they are both of positive charged molecules, but why they have different effect on membrane? To answer the questions, we use the X-ray based techniques to study how the interaction happened at the liquid surface. We use this C-type gold lead rod and PDC gold lead rod to study their interactions. We found that surfactant coated net rod may change the thickness of the lipid. We found that after interaction, the thickness of the lipid changes, and we can also get the quantitative information about the thickness of lipid. For polyelectrolytes, this remain the same thickness of lipid. So suggest that these two kind of molecular modified gold lead rod cause a different effect to the lipid. Then we can see that for the surfactant coated nanoparticle, the lipid area increased due to the detect of the moleculars. They were insert the lipid, but for this one, they didn't change a lot for the area of lipid. We can see the mechanism for these molecules. They were lipid and insert the structures, but these are stable on the surface. Then we use the simulation to study the two kind of nanorods. For surfactant, we can see the ligand may penetrate the liquid penetrate the lipid. This is lipid. This is the C-type molecules. They can penetrate these lipids and change the structures. And this is the polyelectrolytes. The ligand remained at the nanoparticle and the lipid interface. So they didn't change the structure of lipid and cause the next cytotoxicities. Next, I will introduce the application of single-cell imaging for nanomaterial and cell interactions. As we know just now, a nanomaterial are coated by proteins before they enter the cells. So we have a question whether we can detect the protein, the absorbed protein when they go into cells. Other when the protein contains some sulfide, we use this system as an example. We use a gold nanorods coated proteins, protein-coated nanorods to study the interaction with the cells. We found that after interaction with cells, the element imaging for gold and sulfur increased with time. So this, based on X-ray fluorescence imaging, can show the direct evidence for nano and protein corona going inside the cells together. Moreover, we can use the STIGSOM to study high resolution for nanoparticles, especially for some particle hardly captured by TM. For example, we use this kind of chemical, this kind of nanoparticle. This particle only contains one element, one metal elements. It's hardly to detect them by TM because the contrast is very low for TM. So we think out whether we can use the element-based imaging to detect this nanoparticle when they uptake by they are uptaken by macrophage. We found that this method can detect can be used to imagine elements based on the pre-age and on the age element absorption, X-ray absorption efficiency. So based on these properties, we can detect the different energy, the image at a different energy. Based on the pre-age and on the age, we can see the different signals for the samples. Then we subtract the signals, we get the distribution of elements. So we using this method to study different time at different time the distribution of elements inside the cells. So this method can successfully detect the some challenging nanomaterials inside the cells. Moreover, we can use the method to do 3D imaging to based on XRF and STIGSOM to see the elements inside of the specific organelles. Because we use the STIGSOM and the CDI, we can detect the electron density of the samples. Different organelles, some such as the nucleus, endosome, lysosome, mitochondria, they have different electronic densities, so we can distinguish different organelles. Then we based on X-ray XRF imaging, we can show the distribution elements. Combined together, we can know the colocalization of elements at the 3D imaging, at the 3D level to show the distribution. Moreover, we study other nanomaterial inside cells. We can use this method, STIGSOM, to study some matter containing the nanocarrier to study how they target the tumor cells. Based on gadolinium elements, we can see the tumor cell uptaken the particle with time. If we use some inhibitor for the receptors, we can find that the uptaken is inhibited. So suggest that the uptaken is of the particle is via some receptors. Moreover, we also use the high resolution imaging to detect what happened inside the cells. Other window, when nanoparticles go inside the cells, they are different. There are several different chemical reactions. For example, some redox of the particles. There are some chemical coordination of some molecules, such as the lipids. Another, maybe they are oxidized into some oxidation forms, and then they may be dissolved in acidic environment to ions. So to understand what happened to the materials inside the cells, we can use the X-ray absorption spectroscopy and the X-ray imaging to detect the reactions in vivo. Other window, several nanoparticles are considered to be toxic to cells and to human beings. However, the chemical reaction mechanism is still not clear. To answer the question, we use the X-ray based imaging and the X-ray spectroscopy to study the chemical reactions inside the cells. This is the nano-CT microscopy. This system, we can see X-ray go this way and sample is here. After the penetration, we can get the rotation information and get a reconstruction of sample, get a 3D imaging of the single cell. And we treated the monocyte cell with the cell particle at different time points and get a sample, then do 3D imaging for the cell particle inside cells. We can see at the beginning, there are very few signals or matters. Here I want to highlight that the red sample is some gold particle to label the sample for images. It's not from cell. We can see at the 24 hours, there are high accumulation of cell particle inside cell. With time, the signal decrease with time. This is the reconstruction imaging. We see the uptake reach the maximum at 24 hours. With time, after uptake, the cell will degrade and excreted by the cells. So the signal inside the cells decreased. Based on X-ray imaging, this is also another aspect of the nano-CT. We see this is uptake time, this is the remove time. The cell signal decrease and this have a high accumulation. Then what happened to the cell? We use the X-ray absorption. We see the time-dependent chemical reaction to cell. We can see at the beginning, the cell remain as the elemental cell. With time, the cell change to the cell oxide and become to cell ions and cell sulfide. So other than the molecular, some protein and structured proteins contain the sulfide. This is cell dissolved cell may target the proteins and damage cell structure and cause toxic. So this work study shows the reason why cell particle induce the cytotoxicity to monocyte cells from the chemical aspects. Moreover, we can use the nano-CT 3D imaging to study how some nano medicine interacts with the bacteria. Other we know some bacterial structure is bacteria can develop a resistance when they are treated by the antibiotics. So nanomaterial have an advantage to damage the structure of bacteria. So in this work, we developed our nanomedicine that can target the cell wall of the bacteria and damage structure. We can, from nano-CT, imagine the inner content will be released and the structure will damaged by the particle. This blue one is the nanoparticle can directly target the surface and damage the structures. So this nano-CT show very powerful information about how nanomaterial interact with the cells and the bacteria. Moreover, we can use the X-ray based imaging to study what happened to the cells when they are degraded inside the cells. Our previous work showed that bismuth sulfate, the nanorod, is hard to be degraded and accumulated in the liver for very long time. So to help the degradation and metabolism, we develop the particle into our small nanodots. We change the size and change the composition of the particle. We found that these nanodots can be quickly removed from the liver. So what is the chemical reaction and the mechanism? To study the mechanism, we use the X-ray absorption to study time-dependent chemical reaction inside the cells and in some environment. We expose the cells to these nanodots. We found that at the beginning, the nanoparticle contain the copper and the bismuth. So we use the copper. For example, we found at the beginning the copper is aggregated inside the cells. This suggests that they are triggered by some vesicles. And also they have a good nucleation with sulfite. With time, the aggregation dots will be disappeared and they are distributed homogeneous. For longer time, the copper decreases the concentrations inside cells. So suggest that the copper particle inside the element of copper in the particle may be removed by the cells. And with time, we also detect the chemical forms of copper. We found at the beginning the copper is the valence 1. With time, the will be oxidized from valence 2. So these two methods can well explain how the nanodots, what happened to the nanodots in chemical ways, and explain how this particle is metabolized by our body. Finally, I would like to show how these techniques study the chemical phase of nanomaterial in vivo. As we know, some nanomaterial, when the nanomaterial was injected in the blood, they will target some important clearance tissues such as the liver and the spleen. So we use the X-ray based techniques such as X-ray fluorescence imaging and X-ZAPs to study the details. This is one example of how MOS2 nanoparticle are accumulated in the liver and removed in the liver. We use the X-ray when the particle injected in the blood, they are quickly go to the liver. So they go to the liver because this medical particle have the metal MO elements. So we use the X-ray XRF imaging. We can see the distribution of elements of MO in the liver. Then we use the high resolution mode to detect the MO. We found the MO have some specific distribution in specific cells. We use the IF in fluorescence immune fluorescence to detect the macrophage inside the liver. Found that MO have a very high nucleation of the copper cell. This is a kind of macrophage cell. So they have very high nucleation at the beginning. With time, we found that the distribution, co-distribution of macrophage will be disappear. They will have more distribution with the liver cells. So what happened to the MOS2 nanomaterials? We use the ZAPs X-ray absorption spectra to detect what happened to the liver with time. At the beginning, we found that the MO have a valence 4. With more time, the valence will be changed to valence MO valence 5 and valence 6. Other we know the MO element is important for the enzyme, MO enzyme inside the liver. they enable, for example, AOX or XOR. There are two kind of metabolism enzyme, and including MO elements. So we found that after exposure to the nanoparticle, the level, RPDR, MR level of these two molecules increase with time. So suggests that MO may be transformed to some bioavailable molecules. These two enzymes have some MO cofactors. So we, based on this molecular biology evidence and X-ray based evidence, we can see that when MOF2 nanosheets were injected in the blood, they go to the liver, then they will be metabolism and degraded into MOO4, this kind of high valence forms. Then this kind of form may transform MO factors into the MO enzyme. There will be, this is one new ways for the degradation and the transformation of nanomaterials. It's a new phase, new way for the phase of nanomaterial. They can become a part of the molecules after the degradation and transformed. So this work, so we're go to the summary. We use the X-ray based techniques to study how nanomaterial interact with our biology system at a molecular level, cellular level, and the tissue and the whole body level. These techniques can show very important structural information. To, for example, can detect the protein signal structure, can show the lipid structure, and this information can show what happened to nano biology at the beginning. Events. Later, when the nanoparticle go inside the cells, there are some kind of physical and chemical reactions to nanomaterials. For example, some redox, kinetic, and degradation and transformation and biotransformations. During these chemical reactions, X-ray based imaging and X-ray absorption based on spectroscopy can detect chemical reaction in situ. This in situ where well explain the chemical fates of the particles inside the cells and in the tissue and in the whole body. So we think this combined of X-ray imaging and the liquid environment nano-bio interface techniques can show very detailed information for nanomaterial interaction with our body and the system. These are also powerful for the development of nanomedicine in the future. And in the future, not only for the performance of nanomaterials, but also for the safety evaluation. Moreover, these techniques are not only powerful for the nano and bio interaction, they are very powerful for the application in the environment science. They can be also used to study the particles, the haze particles in the air pollution. We also do some work to study how the metals are distributed in the particles, in a micrometer sized particles, to know how the particle induce the oxidative stress and toxic to cells. So I think these powerful techniques will be helpful for our study, not for nanobiologists, but also for scientists in environmental science. And I would like to thank my collaboration with my colleague in Institute of Hydrology, National Center for Nanoscience and Technology, Professor Chun Chen, Professor Xiaochun Wu, and Professor Yuniang Zhao. And I also like to thank my collaboration with the Synchrotron facility in China and from US and Canada. They provide a very plenty beam time and a very instructive powerful techniques for our study. So I would like some fundings from China and the Hefei Science Centers. Finally, I will thank you for your attention.
Thank you very much, Professor Wang. That was an excellent introduction to what obviously is a very complex biological systems in which you're working. I would now invite people to ask questions. I would also invite you to turn on your cameras if you wish, so that we can see who's actually asking the questions. Part of the purpose of these lecture series is for you to get the opportunity to perhaps get to know people around the world working in other areas. So you can ask your questions either using the chat function, or you can open your microphone. And I see that we have a question from Ian Oliver in the UK, who asks, your data shows that the nanoparticles can penetrate cells. Once they have penetrated cells, do they tend to remain inside them, or are they still mobile and reactive?
Thank you, Professor Evans. Thank you for this question. See the nanoparticle can penetrate. Oh, it's very good questions. It depends on the cell type and the type of nanomaterials. For example, as we know, for some carbon nanomaterials, and when they are when they are uptaken by the macrophage, most time they are remain in the cells. For some metal particles, they are chemical reactive. For example, for copper and silver, they will be moved. They will be degraded. They will be removed by the cells. Moreover, some particle may be transformed from one place to another. One of our previous work showed that if we coated the particle with some surfactant molecules, we coated the gold rod with the surfactant molecule, such as CTAP. This molecule can damage the membrane of the nanosome. Then in this case, the CTAP gold rod may transfer from nanosome to mitochondria. So this particle moved from one place to another inside cells. So the answer is that it depends on the cell type and the material type. Thank you for the question.
Other questions for our speaker today? Maybe while you're thinking about that, let me ask you one, Liming. A lot of what you've shown us today is experimental work where you can, prepare your sample and then directly take it to the facility. But if we think about extending this to more environmental type samples, does sample preservation become an issue? In other words, how long would you need, how long could you wait before you analyze a sample? And given the limited number of facilities that have the synchrotron type facilities that you're talking about, how does that play into ability to analyze real world samples?
Yeah, very good question, Professor Ivan. For as we know, the beam time for synchronous radiation is precious. For for different sample, we have different way to prepare. For X-ray based liquid, it didn't chemical reaction is not involved. We can do them real time. Yes, we can take a sample and prepare the lipid on the trough surface that we can do at once. We no need to care about the pre-sample. For some cell and nanomaterial interactions, it actually challenges. For example, for gold nanorods interact with cells. Gold is inert, so maybe not the problem. We can, if we incubate the cells with a particle, and then dredge them and store them in air-free tubes or air-free containers. Okay, and then we take a sample for X-ray imaging and detections, even no problem. But for servo, it also questions. It actually questions because servo is chemical reactions. Yeah, we just try our best to shorten the time between our experimental time and sample prepare time, because with longer time, the servo may interact with the monoclonals and some things. Actually, we one best way that we dry samples and put them in air-free system and in cool system. And in the future, we want to do some improvement. For example, we want to try to develop some system to quickly dry the samples, maybe in several hours or several minutes to dry the samples. That is a better way to get the real information. Thank you.
Okay, thank you. Again, I'll invite other questions, but if there's no question at the moment, I have another one for you. So any questions for Liming?
Yeah.
If not, this actually follows a little bit on my last question. When we get into real world samples, so for example, some of your work on inflammation or drug transfer and so on, in the real world, people might have other sources of nanoparticles that might interfere, I guess I would say, with some of the signals. And I guess my question is really about the complexity of samples and how that affects your ability to separate out specific signals for some of the nanoparticles that you're looking for.
Pardon, may I ask you a question about nanoplastics?
Yeah, not about nanoplastics, but about, so for example, if you're interested in looking at the effect of, say, a drug delivery that you're using a nanoparticle carrier for, right?
Yes.
And in your work where you have that in a sort of isolated system, you can see very clearly the signals and watch the transformation or the translocation of the drug, that sort of thing. But in a real world individual, where if I'm taking that drug, let's say it's a platinum drug or something like that, but I might have other sources of materials that might interfere with those signals that you're seeing. And so I'm just wondering about how complex samples affect your ability to actually trace some of these processes that you're watching.
Yeah. Actually, the signal pathway is too complex. But I think it also it may work. I think it works for some signal pathway. For example, usually some nanoparticle may be uptaken by macrophage by some cell receptors. Yes. In this case, you want to verify which kind of receptor are involved in the uptaken. So you want to study if you want to study that, you can use some antibody and some small molecular inhibitors to block block the surface of macrophage. Then you can see very less of the uptake or distribution of the metal elements inside the tissue macrophage. Sorry, I'm not sure I can steer. Another way is that there's a American company develop elements, rare earth elements based antibody. How to say? The antibody contains some rare earth elements. You can use the antibody to block the specific molecular in the target cells. You can not only see the particle elements, but also see the rare earths, just the receptors, to see whether they are colocalization or other nucleation. I think this is very interesting to see what happened to cell in vivo.
Yeah, that makes a lot of sense. Okay, thank you.
Thank you.
So other questions for a speaker? This is your last chance to get in and have your questions answered. So I have a question from Peng. Thanks for your excellent report. And I wonder to know if synchrotron radiation technology could be used to observe the interaction between proteins and nanoparticles on bacterial cell membranes, and how to determine which protein on the membrane is bound to the nanoparticle.
Yeah, well, thank you for this question. Actually, I am doing this study now, and it's a very interesting topic. Just as I mentioned just now, because first we need to confirm the species resolution of the X-ray images. Yes, if the how say, the native the best resolution now may be rated to 2 nanometer resolution. I think it is possible to detect a single protein on the bacteria. And I mentioned that US company we can buy some rare earth element modified antibody, and that antibody can target the surface of bacteria. So first you label the surface with the rare earth elements, then you use your metal nanoparticle interact with the bacterial surface to see whether they have a good colocalization. If you have good colocalization, they can see this protein may be the target proteins. Of course, X-ray based techniques can show the direct evidence for the interaction. I think another way you can use some biochemical ways, such as SPR and proteomics. Proteomics can be show the direct interaction. Now we are doing a similar work. We found some material that can highly target the bacterial membrane. We don't know what kind of protein, so we can separate the cell wall or cell membrane, bacterial membrane, bacterial wall proteins, and break them into proteins. Then incubate the protein interact with the particle to see what kind of protein are enriched on surface of particle. If this first way you can find your target protein, then you use the metal label to specific protein. So I think the proteomics and the X-ray imaging, they can be combined together to show how they interact with the particle, with the proteins. I'm not sure whether this answer can well address your concerns, Professor Peng.
Well, I would also remind people that you're more than welcome to get in touch with our speakers. And if you need to get an email address, you can contact Julia Colley, who's our events coordinator, and she will put you in touch with speakers if you want to follow up on anything. We have a question from Kelly Evans, who asks, thank you for your talk. I'm wondering if nanoparticles are more likely to bind to proteins located outside the cell membrane compared to those located inside the membrane.
Thank you for your question, Dr. Evans. More likely to bind. Oh, this question, a very good question. And I just talk, I just answered to Dr. Peng. I think we need to use some biochemical tools to study which protein is better or either have a stronger affinity to the nanomaterials. I think, for example, if we want to study the outside the protein, we just outside the membrane, we can first to detect, use proteomics to detect what kind of protein prefer to absorb on surface. For the inside the membrane, we can break cells into pieces and go to the cell lysis proteins interact with the materials. We get another part of proteomics. So from the two parts of proteomics, we get different specific proteins. Then we use some method like SPR and BLI and MST, other method to detect the binding affinities between nanomaterial and the different proteins. So that crystal time is also dependent on the material type, also on the protein type. Sorry. Thank you. I'm sure my answer is can well address your concern.
I think that answers the question. Okay, I think given the time, we'll call a halt to the questions. Again, as I said, you're more than welcome to get in touch with our speaker and ask additional questions by email after the talk. I also would remind you that these lectures are recorded, and so you can always go back and look at them from our website on our YouTube channel. If you have any trouble getting access to the recorded talks, just get in touch with either myself or with Julia Colley, and we'll find a way to make sure that you can see them. And I just want to give a bit of a preview of our talk for next week. Next week, we will have Jason White from the Connecticut Agricultural Experimental Station. We're going to change the nature of the talks, where we're going to get more into applications of nanoparticles. And Jason will be talking about nanoparticle-enabled agriculture next week. So I would invite you all to come back and hear that talk. That will also be a very exciting talk. And I want to just conclude today by once again thanking you, Professor Wang, for this really interesting talk. We really appreciate you taking the time to speak with us, and you've certainly introduced an area that's going to be very important as we start to look at using nanoparticles in a lot of different biological systems. So thank you again for today. And thank you all for being part of the lecture today. We'll see you next week.
Thank you, Professor Evans. Yeah.
Bye for now.
See you next time. Bye.